behalf of Maria, which unfortunately could not make it come. Of course, we would like to thank the organizers for the invitation to discuss the role of telomerase and telomeres in aging. So, yeah, I start my presentation with one of the simplest protocols of aging. There is culturing cells in vitro. So, like a normal cell, mortal cell, you have a limited number of divisions until you reach senescence, and the immortal cell you can divide almost forever. So, I mean, some of these pathways that distinguish between these two conditions could be useful to have an impact also on aging in more complex systems. Also from here we can, define the notion of immortality. We can be immortal at least at the cellular level. And why aging research? Basically because in the last decades there's been a huge increase in the average age of death. And of course we have to pay a price for it. So the probability of developing a cancer is around 1 in 2 in men, 1 in 3 in women. And cardiovascular disease is more than 50% the lifetime risk in men and around 40% in women. So if we somehow can control and try to decrease these risks, it will be important things that we have to address in the future. So today I will discuss the telomeres as aging markers and also the role of telomerase expression in tissue fitness. So the first part, what telomeres are. Telomeres are repetitive DNA sequence that are at the end of the chromosomes. And these sequences are protected by a number of proteins called shelterings, and these varies. The size of telomeres vary a lot between different species, such as 10 kilobytes in humans, around 40 in mice. And what they do is mainly protect the chromosomal endings. There's a complex of proteins, telomerase, that adds this repetitive sequence. They could be extended by this protein complex. However, the somatic tissues are not positive for telomerase. So, can pass here. So they get shortened in each cell division to the end replication problem. So the polymerase cannot deal with these gaps, and so they get shortened with cell division. And the problem is that when they get too short, they are recognized as the double-strand breaks, and so the short telomeres could lead to cell cycle arrest, senescence, or apoptosis. So, the role of telomerase and telomeres in chromosomal protection, cancer, and aging was first assessed from these mice models that we produced, that were first produced when Maria was doing the postdoctoral, and then we characterized them in Madrid as well. So the telomerase deficient mice, third knockout, they present stem cell dysfunction, and they also present less cancer. And on the other hand, the mice overexpressing telomerase, they present a better tissue fitness and slightly more cancer. And when they cross these telomerase overexpressing mice with these three tumor suppressors, so P16 that you heard in the last talk, ARF and P53, we could get an overall increase in lifespan of 40%. So mice expressing telomerase with tumor suppressors, this leads to significant lifespan extension. This is the same equivalent in humans to extend a healthy life until around 100 years, although the maximum lifespan did not change. So increase the years of healthy life. So, returning to the telomeres as aging markers, what we know is that these telomeres, they get short with age. This is in men and women, so as long as the time passes, they get short. And this is telomeres measured in the peripheral blood. Probably more important than this mean telomere length is the percentage of short telomeres that also increase with age. So this is just the observation. So the first thing that we thought was if you could use this telomere size to predict lifespan. And so what we did was we measured the telomere length at 4, 8, 12, and 25 months old mice. The same mouse we measured during the entire life. And we could observe that first, the telomeres get shortened, and there's an increase in the percentage of short telomeres here. It is in the different time points. And probably more important is that you cannot see. What is written here is that the increase in the percentage of short telomeres per month correlates with the realized lifespan. So just scheme. So different mice, they have different rates of increase of short telomeres, and from this correlation, you could see that the mice that gain more short telomeres, they die before. So it's a good indicator of the final lifespan. So the question that we decided to address after was, if we change the telomere dynamics, can we modulate life and health? And for this, I go to the next part of my talk, that is the role of telomerase in tissue fitness. So we thought about the direct expression of telomerase in adult tissues, because the other mice was expressing telomerase from the germline, so we were also interested if we could revert the aging process. And for this, we pick a gene therapy approach. Why? Because mainly because it was easy, and now there's very good strategies to express telomerase. We choose the AV9 expression of telomerase. AV9 is an adeno-associated virus, and why AV9? So first, because they infect many tissues, they are non-integrative, and they are non-pathogenic. And this is a paper from 2006 where we can see that it affects several tissues: liver, heart, muscle, pancreas, almost everywhere. And so these first slides were just the AV9 with GFP that we use as a reporter. You can see mice infected with GFP. This is, controls with telomerase, so they don't have this direct GFP signal. You can see that almost every tissue is infected. This is just separated muscle, brain, heart, liver. This is immunohistochemistry against GFP. More or less the same tissues. We have around 20 to 60% of the cells infected. So we are expecting to have the same percentage of cells when we use the telomerase virus. So unfortunately, we don't have a good antibody for telomerase. The good antibody that we can use in immunohistochemistry. So we test the telomerase expression first through mRNA levels. So you can see an increase comparing to the controls. Here they go. The controls that don't have telomerase or FU. You could see also by Western blot in different tissues, the quantification of the Western blot, and really more important, using trappist, so these bands. This means that the telomerase is active, so they say to see telomerase activity in vitro, and this is extracts from lung from 2-year-old mice infected with telomerase, so it is free mice or GFP. So you see that there's only activity in the telomerase infected mice. So with this ectopic expression of telomerase with AV9 leads to stable protein expression for at least 8 months in vivo. So after we were interested to see, this is a simplified protocol. So it was in fact GFP or telomerase in 1-year-old and 2-year-old mice, and see if we can revert some of the age phenotypes, if we can call them phenotypes. One of them was bone density. So we know that bone density decreases with age. In this sense, we compare both groups of mice. So we did not follow the same mice. So we compare 1-year-old with 2-year-old mice. This is the p-value for the age. It's 2A and over. So we can see that there's a decrease, a significant decrease with age, and this could be reverted through telomerase expression. So these mice are expressing telomerase for 6 months, for 3 months. You can see that this reverts the bone density. The second part is subcutaneous fat layer, so this decreases with age, and this could be also reverted only in the 2-year-old mice. Some of these conditions can be reverted only when you are too old. 1-year-old mice, they have more or less a normal subcutaneous fat layer. Insulin levels were also lower in the mice infected with the telomerase virus. IGF-1 is also known to decrease with age and was rescued after telomerase expression. And the glucose tolerance, the mice infected with telomerase, although it's not significant, they also present a better tolerance to glucose. So, in 2011, appeared a paper, a Nature paper, that demonstrated that telomerase could be playing a role through the mitochondria in stem cell exhaustion and tissue impairment. This was one of the questions in the telomerase field, was what telomerase is doing in post-mitotic tissues. I mean, it's easy to understand that in a tissue that divides, we lose telomeres. However, in the post-mitotic tissue, it's quite difficult to understand if telomerase could be playing a role there. So what they observed is that, I mean, telomeres can get shortened in post-mitotic tissues, and this impacts in the mitochondria, and this could lead to some of the phenotypes observed in the third knockout mice. And so we checked for some targets. Many of these is also not significant, but you could observe that, like, PGC1 alpha or ATP synthetase in this case decrease between the 1 and the 2-year-old, and could be partially rescued through telomerase expression. So seemingly there's an impact in the mitochondrial function. More recently, we were looking to have a collaboration that was also interested to study the metabolomic profile of these mice. So it's, what you do is just collect serum from these mice and do a metabolic profile. We identified many metabolites, many lipids, creatinine, methionine, that seemingly go down with age, others go up. And this is a principal component analysis. I mean, the information that you can take from here is that this is the 1-year-old GFP and the 2-year-old. What you can see is that the 2-year-old has a shift in these metabolites. I mean, they change with age. Some of the metabolites go down, others go up, but there's a difference with age. When you compare this with the telomerase, you can see that they are much closer to the 1-year-old condition. This is serum from the mice infected with telomerase. So it's just another proof that these mice look younger. So continuing characterizing these mice, this is post-mortem analysis. So mice, they have more or less the same inflammatory pathologies. However, the telomerase infected mice, none of them present fatty liver. I mean, these mice were in normal conditions, they are black 6, but you know that the black 6, they tend to be obese, so 10% of control mice present fatty liver, and none in the telomerase. We also did some neuromuscular tests. The thyroid test, it's just, you put the mice in the virus, it's just to test the neuromuscular strength. This is another assay that we get better results with very old mice, and we could see only difference in the 1-year-old group, 11 months post-in injection. So you can see that there's a decrease of this capacity with age, and the telomerase mice were also, they scored higher in this test. They also scored higher in the rotarod, and this measure also coordination and balance. And then after all of this, we were also interested to see if this could impact the lifespan, and indeed it impacts. So in this case, we compare as well with the control, so non-treated mice, and you could see around 24% of increase in the mean lifespan, comparing the telomerase with the GFP, a little bit less with the control, I mean non-infected mice, but it's significant in both comparisons. In the 1-year-old group or in the 2-year-old group. In the 2-year-old group, I mean, it was around 10%. Probably we don't have so many to change in this case, but anyway, there was a significant change. And probably more important than this was when we characterized, I mean, the pathological analysis of these mice. So in both groups, they present more or less the same amount of cancer, so 66 to 64 infections and other pathologies related with age. So when we stratified the cancer, they present more or less the same percentage of lymphomas, adenomas, and this 2% is only one mice that presented adenocarcinoma, and this is important because telomerase is, I mean, when we have telomerase expression from the germline, this leads to more cancer. And also there's many links that associate telomerase with cancer. So, but seemingly when we express telomerase late in life, we cannot recapitulate this cancer phenotype. So this is just a small scheme. So the short telomeres lead to degenerative pathologies. I'm not saying that this is the only cause of aging. I mean, when you have aging, you have an increase in short telomeres, and somehow they can explain the aging process. And telomerase re-expression can rescue the short telomeres. I mean, I did not show these results, but we see a rescue of short telomeres, and there's an increase of health span and increased lifespan. And just to finish, I will pass in a few in the recent bibliography, because we are not working alone on this, fortunately. So in 2011, there was a paper from Devino and Jaskeliov. So they use an accelerated model of aging, the G430R. So these mice, they don't have telomerase, and you went to the 4th generation, and they are telomerase off. And when they activate telomerase with tamoxifen, they observe telomere extension, reduced DNA damage, and reversal of the degenerative pathologies. And it's just a representative they also saw in, I mean, brain is not completely post-mitotic, but for instance, the brain, they can see after tamoxifen induction, 4 weeks, the brain is more or less similar to a G0, so this is a control mouse. More recent papers are this one in Aiton from 2012. They also use a telomerase activator. It seems to be protect from the progression of ALS. In 2011, there was also a group demonstrating that intrahippocampoid infusion of recombinant adenovirus expressing telomerase impacting neurogenesis and brain protection. And also 2011, there was some viral group demonstrating that viral telomerase delivery to the spinal cord cells increased the regenerative potential. So I mean, seemingly there's, there's many groups working with telomerase in tissue regeneration. It seems that something is happening, and we just have to see how to use it. So, and I finished. So the acknowledgements, of course, go to Maria and our collaborators, Fatima and Edward, that produced the virus, our pathologist, Joana Flores, and to the CNIO units. And this is CNIO in Madrid, if you want to visit it. And thanks for your attention, and I'll be happy to answer to your questions if you have some.
Hi, that was a great talk. In mice, the telomere length, of course, is quite large, and you showed one of the slides. You showed that there was a 40% lifespan extension in mice with telomerase expression, overexpression. Is that right? They get 40% benefit, but that was in cancer control mice, right? The 40% lifespan extension
was in telomerase together with tumor suppressors.
Yeah, with tumor suppressors. Yes. So, because otherwise, with just telomerase, you get an increase.
Telomerase, no, you have the same lifespan. I mean, you have the same mean lifespan, but the mice that don't have, you have the same mean lifespan, even considering that the mice have much more cancer. But the mice that do not have cancer are the longest lived. Understand? No.
Yes, yes, yeah, no, I understood that.
The telomerase group.
Yeah.
So I mean, the telomerase group mice, they have more cancer, and the same mean lifespan as the mice that do not express telomerase. And in the telomerase group, the mice that do not develop cancer are the longest lived.
Yeah, no, I understood.
Both groups.
Yeah, absolutely. But the idea is to increase telomere length, right? I mean, with the telomerase expression.
We believe more that is protecting from short telomeres.
Protecting from short telomeres.
Yes, and
So
probably there's many new work on these, and there's links with telomerase and the Wnt pathway with other pathways, and these with the mitochondria. I mean, post-mitotic tissue. So it's difficult to believe that a post-mitotic tissue such as the heart, for instance, or that you can see an actual extension of telomere length. Probably can see some from protection of telomere shortening, but not a real extension of telomeres, because you need the cells to divide for the telomeres to get to extend. On the other way, if you protect from short telomeres, if you measure telomere length, of course they look longer, but because you have less short telomeres, not really you have an actual extension of telomeres. And so you just a part of the short telomeres, if you take them, of course the mean will look longer. It's It's not the real extension, it's just a protection of short telomers.
Other questions? Edward?
I had a question about the next slide, the action. So we were talking about those super mice that live 40% longer than their controls.
The next one, I mean, this is basic research, and the next plan is, of course, trying to find a way to use this. And there's many telomerase activators now. Also, there's many companies selling telomerase activators. Some of them were characterized in our lab, and probably this is not the best way that we can go in the future. What we are now planning to do is go at the tissue level to see how telomerase can be used at specific organs. We can counteract this age decline in specific organs. I mean, we don't believe, also like in the T16 presentation that was just before mine, seemingly different organs, they have different rates of aging and depend from different circuitries. So probably can use telomerase in some organs that we know that age due to telomere shortening in normal conditions. So eyes, bone marrow, or any other organ. And I don't think, I mean, this was a good way to show telomerase could be used in normal conditions and their impacts on lifespan, but now we have to understand at the organ level how it can be used.
So just perhaps to be a bit more precise, there are people selling TA65 and other things to extend the telomeres, and they based the sterility argument on some studies that were made for HIV patients. I guess the safety requests are not the same for an HIV patient and the person who doesn't have a pathology.
They are selling TA65, and there's humans taking TA65, and we were writing TA65 in the lab in the paper that was published in 2011, I think. I'm the author and with other people, but we don't see any lifespan extension. We see some health span rescue. I mean, some specific. The problem is the biodistribution, because I mean, you have a huge increase of telomerase expression in the liver, but not much expression in any of other organs. And also the mechanism. It's not a direct activation of telomerase. It's a secondary, it's through the pathway, probably through MYC. So I would say that we need more research on TA65. We saw some rescue of this aging phenotype. I mean, glucose tolerance, these mice, they are much better than the ones with the virus. With TA65, they have a very, very good glucose tolerance, but they do not live longer. The cancer was also more or less the same. I mean, a little bit higher in the TA65 group, but it was not significant. I would say that it was the same. But I mean, I think we need more research on this.
What mice were these tested in the cancer?
These mice.
Knockout mice, the TA65.
We're in the same. We're black 6, 6.7 black 6. But we're not the same from this. We're not the same from this group because we had to buy. We didn't have mice with this. We want to start it immediately in 2-year-old mice, so we bought them, and they were not exactly the same strain as these mice.
Yeah. So we did also lifespan with 2 doses of TA65 in these black 6 by C3H mice, and found no effect on lifespan.
And
No effect on lifespan.
no, we did not.
Has anybody seen any human data for the TA65 that is not done by the company itself?
There's a paper. There's a paper in 2010 that, from Ireland, in Ireland, and they characterize more or less the people it's taking. But I mean, there's not really a control group in the paper. I mean, it has to be careful to see that it is there. And there's this HIV paper. The HIV paper is not exactly the same. It's T82. I mean, we believe that it's the same compound, but we cannot